Good morning everyone. Good morning, good morning. What is happening? Okay. Okay. Good morning everyone. Good morning. Good morning. Okay. So friends, today we were started discussing about the biochemistry. Okay. So friends, listen to me carefully and what are the various topics that I am going to cover in this particular chapter of the biochemistry. Okay. Everyone follow me correctly friends. In the chapter of biochemistry, we will be majorly covering the carbohydrate metabolism. I will cover 5 very important topics where we get the questions for very sure. Okay, we are discussing about the carbohydrate metabolism. We are discussing amino acids and proteins. Okay, and we discuss enzymes and its kinetics. Okay, along with the inhibition. Okay, so compulsory we will read the process of enzyme inhibition as well. Okay, along with the enzyme inhibition, we will study the RC plot. Okay, and I will explain you the beta oxidation also. That is, beta oxidation is what? Fatty acid metabolism. Okay, so these are the 5 to 6 concepts that we are going to discuss in the chapter of biochemistry. Okay. Okay, so now let's start the carbohydrate metabolism. Okay. Good morning everyone. See friends, if you look into the carbohydrate metabolism, I'm talking about the September 2022 paper. I'm just talking about this paper only. Okay, we got one part B question directly from glycolysis. Okay, directly, but it's a very simple question. I can use the word very logical. Okay, and we got another direct part C question directly from the glycolysis regulation. Actually friends, both the part B and part C question actually asked from the regulation of the glycolysis. A very simple and very straightforward question, but it's a kind of a twisted question. It's a straightforward question, but it's a twisted question. And what is the another part C question we got? Another part C question we got from the Krebs cycle intermediates. Okay, which is also a direct question, but most of the people will try to avoid this particular question when they are studying, because most of the people will think that that information is not that much important. So that's the reason I am just requesting all of you, when you are reading the carbohydrate metabolism, even this time also you get, see, we at least we have to believe that we will get either in the part B or in the part C question. So I want every student has to read a very thoroughly. First thing is the glycolysis and its regulation. Do not forget the regulation. Then Krebs cycle and its regulation. Okay, electron transport chain, and once again we got the question is what? Inhibitors. Okay, then followed by oxidative phosphorylation. Okay, these 4 steps I want every one of you to understand. Okay, so friends, before starting the concept, let me tell you one line. In this particular biochemistry, remember my words. Always remember these lines. When you are studying the biochemistry, one point has to strike in your mind. In the biochemistry, how much ever you may study, doesn't matter. If you don't apply your knowledge, trust my words, blindly, you can't answer the question. So biochemistry is always about analysis. Always about analysis. how nicely you are analyzing the question, how nicely you are understanding the question. Based on that only we can decide whether you can answer the question or not. So for that reason, there is a no doubt, you have to remember a lot of concepts. At the same time, remember my words, you have to understand the concept. I will solve this September question and June questions. Now you just imagine, while answering the question, you imagine, you don't need to remember much amount of the information. You all has to understand the information correctly. By that you can solve the questions. So I am repeatedly saying to all of you, while learning the biochemistry, try to understand the concept. You remember the concept, there is no doubt about it, but try to understand. By that you can apply your knowledge. Okay friends, let's quickly start. Carbohydrate metabolism. Friends, how can we define the word metabolism? How can we define the word metabolism? Metabolism means what, friends? A series of two major reactions. What do we call them as? One reaction is called as a catabolism and one reaction is called as anabolism. I want you to remember the simple statements. Friends, when I use a word catabolism, what I am doing, friends, I am always breaking down something. Correct na? When I am using the anabolism, what you have to understand? You have to understand that I am building up always something. I am breaking down something. I am building up something. So now understand everyone. If I want to break down something, all the students follow my words very correctly. Suppose example, whatever the food I take, what my food contains, everyone please continue. My food contain polysaccharides and the food contain proteins and the food contain lipids. Correct na? Food contain lipids. Now you tell me, friends, why we consume the food? Our ultimate goal is what? From the food only we can get the energy. In order to get the abundant amount of energy, friends, we are going to break down our food molecules. So polysaccharide, proteins as well as the lipids. Correct na? So when we break down them, what we get, friends? We get that energy. Okay, finally, oxidation. What is oxidation here? Breaking down. So breaking down of a large carbohydrates, large fats as well as the large proteins. Good morning. Ultimately what we are going to get, friends? Ultimately we are going to get the energy. So what most of the students will get to understand is that in the catabolism we are not producing the energy. Remember my words, in the catabolism we produce the energy. There is no doubt about it. Now, why we are producing the energy? I told you many times to all of you, 37.2 trillion cells which are present in our body. Remember my words, 37.2 trillion cells present in our body. Everything is under the control of what, friends? Everything is under the control of energy. So every cell require energy whenever it is performing a function. So what let's take an example. Contraction of a muscle. Say an example, transport. Say an example, synthesis of something. For every single activity that is happening inside the cell, there will be a requirement of energy. So what we are doing in the anabolism, we are utilizing the energy. Don't confuse, friends. Where do we produce the energy, friends? We produce the energy in the catabolism. And where we are using that energy, friends, we are using that energy in the, what we can say, that we are using that particular energy in the anabolism. So catabolism and anabolism together only we call, friends, metabolism. Okay, correct na? Friends, every one of you try to understand one important point. Whenever I take a food, I can't get the energy directly. Yes or no? Of course I can't, because whatever the carbohydrates, whatever the proteins, whatever the lipids that I consume, they are in a large form, which means they are always in the complex form. So from the complex molecules we can't get the energy. It is not at all possible. So what should I do? From this particular complex molecule, I want to convert these complex molecules into a very simple molecules. Don't get confused with this point. Okay, digestion and hydrolysis. Okay, so breakdown of a large molecules to the smaller ones which are entering into the bloodstream, right? So first what we have to do, we have to break down the large one into smaller ones. When we break down the larger one into smaller one, friends, it is very easy to allow the process of oxidation. What is oxidation? Breaking down. You all people know that What's the ultimate goal of this particular oxidation? Breaking the large molecules into a simple compounds like a 2 carbon containing compound and 3 carbon containing compound. So this 2 carbon and 3 carbon containing compound can easily enter, where friends, where they can easily enter? They can easily enter into the Krebs cycle as well as the electron transport chain. Okay, this is the most important slide that I want every student has to remember, this particular slide. Okay, there are great chances are there that we can expect the statement related questions from this. So I'm requesting every one of you try to understand every single slide. What are the various stages of catabolism? Catabolism nothing but what, friends? Breakdown. All the students please try to understand what I'm talking here. Proteins, polysaccharides, then followed by the lipids. Correct na? This is what present in our food. First let's talk about the major source of energy, that is the sugars. Understand my words correctly. Whatever the food that we take, is it in a simple form like a monosaccharide or disaccharide? Never ever. Whatever the food that we take, most of the time the food is in which form, friends? The food is in the polysaccharide form. I want every student follow my words correctly. If you listen my lesson correctly and if you apply your knowledge, you can answer the question. So I'm requesting sincerely, understand the lesson correctly. Now polysaccharides break down into oligosaccharide into disaccharide into monosaccharide. When we produce monosaccharide from the monosaccharide can easily enter into the process of cellular respiration, whereas monosaccharides can easily enter into the process of oxidation. So friends, the large polysaccharide we are breaking down into simple monosaccharides like a glucose, fructose, galactose. Friends, you all people know very well, fructose and galactose are what? Fructose and galactose are basically monosaccharides only. Is it, friends? So finally the galactose is also obviously converting into glucose. Fructose is also converting into glucose, because you know very well, most of the cells, they don't use fructose. Most of the cells, they don't use galactose for the process of oxidation. What they will use most of the time? They will use undoubtedly glucose as their major source of energy. So friends, understand my words. Here what is happening, friends? The glucose can break down into what, friends? Pyruvate. Anyway, we will understand in the next slides. Glucose undergo the process of oxidation, which we call it as glycolysis. So glucose undergo the process of glycolysis and it is giving rise to what, friends? Pyruvate. Now all the students remember my words. Pyruvate is how many carbon containing compound? I have told you many times. Pyruvate is a 3 carbon containing compound. We also know that pyruvate never ever enter into the Krebs cycle. That is the reason pyruvate converted into an important intermediate which we can call acetyl CoA, and this acetyl CoA is how many carbon containing compound? 2 carbon containing compound. And this acetyl CoA, friends, entered into the citric acid cycle. And what is our ultimate goal? We are discussing continuously. Our ultimate goal is, friends, production of energy. So it will involve the acetyl CoA. Once it enter into the citric acid cycle, it is involved in the production of ATP, production of FADH2, production of NADH. Don't get confused with that particular point. Now this is what? This is the pathway of, this is the fate or a pathway of polysaccharide. Now all the students listen. Now whatever the food that we consume, friends, even it contains the proteins also. Correct now, friends? And understand my words. Protein can break down into amino acids. And friends, these amino acids undergo which process, friends? Remember my words. Amino acids undergo the process of transamination. We discussed it, right, friends? What is transamination? We are transferring the amine group. Upon transferring the amine group, amino acid can give rise to the pyruvate. Some amino acid directly give rise to the acetyl CoA only. Now once again, what you have to understand, acetyl CoA entered into the citric acid cycle. They produce ATP, FADH2 and NADH, obviously. Now we understand the fate of a protein, we understand the fate of a polysaccharide, and now, friends, try to understand the fate of a lipid. Okay, Lipids can be break down into fatty acids as well as the glycerol. You all people know one point very correctly, friends. Fatty acids undergo what process, friends? Fatty acids undergo the process called as a beta oxidation. Yes or no? Like however, what friends, like however glucose undergo the process of glycolysis, similarly fatty acids undergo the process of beta oxidation. Ultimately what the fatty acid is going to give rise to? Ultimately fatty acid is going to give rise to the acetyl CoA. Don't get confused with this particular point. And friends, this acetyl CoA entered into the citric acid cycle. Once again they produce ATP, NADH and FADH2. Friends, now what is the most common statement I want you to understand here? You have to understand a point that whether it is a protein, whether it is a polysaccharide, whether it is a lipid, what's the common point you have to understand? All these three molecules, understand my words correctly, all these three molecules are finally giving rise to what, friends? Acetyl CoA only. So that is the reason I can give a statement that acetyl CoA can act as a intermediate for all the biomolecules in the Krebs cycle. Understand my words correctly. I can call acetyl CoA is the major intermediate in the citric acid cycle, or acetyl CoA is the major intermediate molecule, can be utilized by the protein, polysaccharide as well as the lipids. Okay. Now friends, one common point everyone has to remember: in the process of glycolysis, in the process of Krebs cycle, in the process of electron transport chain, in the process of oxidative phosphorylation, what are the three substances or three precursors that we use very commonly in all these cycles? We use very commonly ATP, we use very commonly NADH, we use very commonly FADH2. Yes or no? These three are present, not only in the carbohydrate metabolism. Let's take a amino acid metabolism, let's take a nucleic acid metabolism, let's take a lipid metabolism. Wherever you take whichever the metabolism we consider, ATP, NADH and FADH2, these are all the common energy precursor. That is the major reason before studying the metabolism, it is compulsory, my friends. First we have to understand about the energy precursors, compulsory. Okay, let's follow me very correctly. ATP and energy, adenosine triphosphate. Correct now, friends? All the students look into the diagram very correctly. ATP consists of what? A sugar. What sugar? Ribose sugar. Then a base called as adenine. And how many phosphate groups are present, friends? Three phosphate groups are present. We can call monophosphate, we can call diphosphate, we can call triphosphate. Sometimes, friends, instead of using the word mono, di, tri, we use the word, all the students follow my words correctly, we use the word alpha phosphate, beta phosphate as well as the gamma phosphate. Okay, so anything you can use it. So friends, what's the important statement I want every one of you has to remember? How can I produce the ATP? Tell me. When I want to form the ATP, I want to compulsory join the ADP with PI. Then only I can form the ATP. Now the question is very, very clear. The question is, do you think I can join the ADP with PI that much easily, friends? No way. If I want to join, I require 31 kilojoules of energy. Okay, first of all, when the ADP combined with PI, I got the ATP. First of all, friends, how does the ADP is getting formed? ADP is getting formed when the AMP combined with PI, then I will get the ADP. So once again, what I have to understand, once again, in order to produce the ADP also, how many joules of energy I will be requiring, friends? Undoubtedly, I required 31 kilojoules of energy. Don't get confused. In a simple line, what I want to conclude? I want to conclude that if I want to produce ATP from the AMP, I will be requiring 62 kilojoules of energy. Vice versa, if I want to break down ATP, if I want to breakdown of ATP into AMP, how many kilojoules of energy will be released once again? Of course, 62 kilojoules of energy is getting released. Either formation of ATP or the breakdown of ATP. While farming, we have to spend energy. While breaking, we will get the energy because energy is getting released. Is it? Okay. Anyway, we solved the questions. I am only solving little faster. Later, I want you to look into the PPTs. So, you know, right, ATP, you use it in the anabolic reactions, right, friends? ATP can act as an energy storage molecule, and we know that ADP and P, see, coupled with energy requiring reaction, whichever the reaction that require the energy, what is used over there? ATP is used. So, all are ATP only. So when I break down the ATP, when the ATP undergo the process of hydrolysis, what I get, friends? I will get the ADP and PI. Simple point. Now, what I want you people to remember is the structure of coenzyme A. You know, no friends, we call it as the acetyl-CoA. Can you imagine any metabolic pathway without acetyl-CoA? Understand my words. Can you imagine any kind of a metabolic pathway without acetyl-CoA? No, that is not at all possible, right? The coenzyme A is an important precursor molecule in many metabolic pathways. Now, all the students try to understand what does the coenzyme A is made up of. I want you people to remember only what does it actually made up of. That's all. It is made up of phosphorylated ADP. It is made up of pantothenic acid and it is made up of aminoethane thiol. Friends, you people no need to remember any kind of a structures. What I want you to remember is, friends, what does it made up of? What does it composed of? Only you have to remember the composition. You don't need to remember anything extra. One is the phosphorylated ADP, another one is the pantothenic acid, another one is the aminoethane thiol. Don't get confused with that. Okay. So friends, like however we are using the ATP compulsory in all the metabolic reactions, we will use the NADH also compulsory. We use FADH2 also compulsory. Okay. Now remember, friends, NAD plus. Okay. I want you to remember the statements very correctly, friends. You all people know that NAD plus, this is what form? This is the oxidized form. Yes or no? And friends, what about the NADH or NADH plus H plus? This is what do we call it, friends? We call it as the reduced form. All the students follow my words correctly. When you heard the name of a NADH, 2 points every student has to remember. What is the first point you have to remember? NADH can act as a reducing equivalent. What it is acting as? It is acting as the reducing equivalent. And what is the second point I want you to remember? You have to remember that NADH can act as an electron carrier. What, friends? NADH can act as an electron carrier in the electron transport chain. These 2 points are compulsory, you have to remember. And what is the third point you have to remember? Most of the time, this NAD involved in a reaction where there is a production of carbon-oxygen double bond. Where there is a production of carbon-oxygen double bond. In these reactions, this NAD, coenzyme NAD is mostly involved. Don't get confused with that particular point. And friends, this NAD plus, look into the structure. It contains sugar, it contains base. And I want you people to remember compulsory, friends. This nicotinamide structure basically derived from an important vitamin. What is the name of that important vitamin, friends? Niacin. I want every one of you has to remember the 4 points. NADH acting as a reducing equivalent. It is acting as an electron carrier. It is involved in the reactions where there is a production of carbon-oxygen double bond. And next you have to remember nicotinamide ring is derived from niacin. Only 4 points I want you to remember. Same, I am explaining the same 4 points with the FAD also. What do you have to remember? What is the reduced form? FADH2. So friends, how the FADH2 is going to act as? FADH2 friends, it is going to act as a reducing equivalent. First point. Now what's the second point you have to remember? Here I told you that NAD involved in the reactions where there is a carbon oxygen double bond will be produced. Here friends, FADH2 majorly involved in the reaction where there is a production of carbon-carbon double bond. Always remember my words. Of course friends, FADH2 also, like NAD, it is also acting as a electron carrier. And what is the fourth and another very important point friends? Here the ring is basically formed from the riboflavin. Here the ring is basically formed from what? Riboflavin. So FADH2 is the reduced form, FAD is the oxidized form. I want you to remember 4 points about the FAD and 4 points about the NAD+. That is the 4 points I want you people to remember compulsory. Okay. Now we understand that ATP, NADH, FADH2 are involved in every single reaction or in every biomolecule metabolism. That's why we have studied about them. Now let's start studying the first one, carbohydrate metabolism. As we already told you, the carbohydrate metabolism ultimately starting from where? Starting from the glucose. So what I am doing actually, you all know that I am breaking down the glucose. Correct now friends? That's why this process is called as what friends? This process is called as oxidation of glucose. I am breaking the glucose into what? I am breaking the glucose into the pyruvate. Why we have to start only from the glucose? Why can't we start from any polysaccharide? Because you know that friends, polysaccharides can't give the instant energy, and moreover polysaccharides are complex molecule, they can't digest very fast. So that's why first we will break down the complex molecule into simple molecules. From the simple molecules only we will start producing the energy. So the breakdown of that glucose into pyruvate only we can call the glycolysis. Friends, we were studying from 11th standard. Always remember once again without confusion. If you look into the simple step of a glycolysis, what is happening in the glycolysis? Understand everyone. One glucose molecule is how many carbon? 6 carbon containing compound. Friends, the 6 carbon containing compound breaking down into what friends? 2 molecules of pyruvate. Yes or no? Of course yes. It is breaking down into 2 molecules of pyruvate. Each pyruvate is how many carbon? 3 carbon. In the glycolysis we will get 2 pyruvate, 2 NADH, 2 ATP and 2 water molecule. I want you to remember 4 2's. Okay. So one is 2 pyruvate, 2 NADH, 2 ATP and 2 water molecule. Now the point is here, I can't break down the glucose directly into the pyruvate. It is a very difficult process. So what I am doing, if I want to break down the glucose into pyruvate, I will be requiring friends, first I will be requiring lot of enzymes. So how many enzymes are required? I will be requiring nearly 10 enzymes in order to just break down one glucose molecule into pyruvate. So that is the reason there are total 10 steps are involved, right? Okay, now you know now friends, the first and most important step, the moment glucose enter into the cell, glucose has to be converted into glucose 6-phosphate. This kind of questions even you can get in the interviews also. Many people who attended integrated PhD interviews, who especially chosen biochemistry, many times this question was asked in the interviews. The people who qualified IIT JAM, people who qualified TIFA. Okay, remember the question is here, why the glucose has to be converted into glucose 6-phosphate? That is the reason. The questions asked is, what happens if the glucose is not converted into glucose 6-phosphate? That is the question. If the glucose is not converted into glucose 6-phosphate, do you think the glycolysis will continue? Never ever friends. The glycolysis never ever takes place. Yes or no? Of course yes. Now all the students follow my words correctly. Why? Because you all people know that phosphate is what? Negatively charged. We all people know that whether it's a positively charged or negatively charged, doesn't matter. One thing is very common: any charged ion cannot cross the plasma membrane. What, friends? Any charged ion cannot cross the plasma membrane. What is the unique statement you people has to remember, friends? Since if the glucose convert into glucose 6-phosphate, phosphate group cannot cross the membrane, so glucose will be in the cell only, so it can't come back, so it has to undergo the forward reactions only. So obviously glucose is going to be converted into pyruvate. Suppose if the glucose did not convert it into glucose 6-phosphate, there is a chance once again they can come back via GLUT transporters. So that is the reason most of the time glucose has to be converted into glucose 6-phosphate. So what we are doing here, just to the 6th carbon we are adding the phosphate group. So obviously the enzyme is what? A kinase enzyme. Since the glucose is a hexose, that's why we are calling it as the hexokinase enzyme. Don't get confused. Here the phosphate group donated by the ATP. Friends, this is the most important slide where most of the people will get a confusion in the exam. You should always understand the given question, whether they are asking the numerical question or calculation, by considering the entire cellular respiration, or they are asking the question only from the glycolysis or only from the citric acid cycle. If you really understand this, trust my words, you can answer the questions very correctly. Anyway, friends, in our competitive exam, CSIR, GATE or IIT JAM, one point is very common: locations where the reactions are happening. That is extremely important towards the exam point of view, especially in the statement related question. That's why you have to know them. Okay, follow me everyone now. Steps of respiration. Glycolysis. First one. What is happening in the glycolysis? Where there is a breakdown of the glucose into pyruvate. What is the first point you have to remember? The glycolysis undoubtedly taking place in the cytosol. What is the reason? What is the reason the glycolysis has to take place only in the cytosol? Because we discussed that there will be a requirement of the 10 enzymes. Where all those 10 enzymes are present, they are exclusively present in the cytosol only. That is the reason, friends, glycolysis always takes place in the cytosol. All the students follow my words correctly. Now, once there is a production of pyruvate, understand my words. What is happening to the pyruvate? Pyruvate has to convert it into acetyl-CoA. Where the pyruvate is converting into acetyl-CoA? This is also a question. Pyruvate converting into acetyl-CoA in the mitochondrial matrix. Where, friends? In the mitochondrial matrix. Now the question is why? The reason is very simple. If the pyruvate wants to be converted into acetyl-CoA, there will be a requirement of an important enzyme complex. What is the name of that important enzyme complex, friends? We can call it PDH. What do you call it? PDH, pyruvate dehydrogenase complex. So where the pyruvate dehydrogenase complex is present? In the mitochondrial matrix. That's why the conversion of pyruvate to acetyl-CoA. You know what do you call it, that process? What is the name of the process where the pyruvate converted into acetyl-CoA? It is called as pyruvate decarboxylation. So where the pyruvate decarboxylation is occurring? In the mitochondrial matrix. Simple. Now finally acetyl-CoA entered into the citric acid cycle, which is the biggest step of the cellular respiration. And where it is occurring? Undoubtedly in the mitochondrial matrix. What is the reason? The reason is very simple. All the enzymes required for the citric acid cycle present in the matrix. And another very important step, oxidative phosphorylation. Where the oxidative phosphorylation occurring? Please remember, friends, it is occurring between inner membrane to intermembrane space. This is the area where the electron transport chain, and electron transport chain is purely on the inner membrane. There is no doubt about it. And where the oxidative phosphorylation is occurring? It is occurring between the inner membrane to intermembrane space. I don't know what you people do. I want every one of you has to remember all the reactions compulsory. Compulsory you have to remember. Okay. Now wonderful. Let's continue. Now let's start understanding the glycolysis. But one point is very clear. Whatever the food that we are taking, they are all in the polysaccharides. Correct na? You all people know that in the mouth, what is the name of a important enzyme located over there in the mouth? Salivary amylase. What that salivary amylase is doing? This particular salivary amylase break down the polysaccharide into what? A kind of oligo and disaccharide, especially dextrins, maltose. But you know very well, in the stomach there will be no digestion at all. Why? Because there is a presence of HCl. Now we all studied many times, small intestine is the area where the abundant amount of digestion actually takes place, which means whether it is a carbohydrate, protein, a lipid, nucleic acid, whatever it may be, complete digestion always takes place in the small intestine only. And in the small intestine there are a lot of enzymes are there, but specific enzymes we can call brush border enzymes. What enzymes? Brush border enzymes. They are the one which break down the disaccharides into monosaccharides. Anyway friends, along with the small intestinal enzymes, pancreatic amylase and certain enzymes from the liver also, they will also help in complete breaking down of large polysaccharide into simple monosaccharides. What friends? Simple monosaccharides. Don't get confused with that particular point. Okay, follow me correctly now. So finally I broke down the polysaccharide into monosaccharide. Now finally the glucose started the process. Friends, let me tell you one common point, very common point. The moment you heard the name of a glycolysis, what is the common point has to strike in your mind? It is obviously anaerobic process. Correct na? There will be a no oxygen is required. Yes or no? If there is a presence of oxygen, friends, of course, you know, the process never ever occur. What is the reason? Because the 10 enzymes of the glycolysis are, remember this line everyone, the 10 enzymes of the glycolysis, they are sensitive to the oxygen. What friends? They are sensitive to the oxygen. What do you mean by that? Which means if there is a presence of the oxygen, friends, obviously the 10 enzymes are getting deactivated. That is the reason if the process of glycolysis wants to occur, it should happen only in the anaerobic conditions. Correct na? I told you multiple times, what's the ultimate goal of a glucose? To produce 2 pyruvates, to produce 2 ATP, and to produce 2 water molecules, and to produce 2 molecules of NADH, right? But the point is, friends, glucose, see, ultimately we are producing energy, there is no doubt about it. But before producing the energy, first we have to invest something. I told you, right, if I want to do a business, and I want to get the profit, I can't get the profit very immediately. When I will get the profit, first I have to invest. So once my business goes well, along with investment, I will also get the profit. So that is the reason in the glycolysis, ultimate goal is to produce the ATP. But if we want to produce the ATP, friends, first we have to spend ATP. I really don't know what you do. You have to remember 10 enzymes. You have to remember 10 structures also. It is not at all difficult, friends. It is easy. Okay, you have to remember 10 structures also compulsory. So 1 to 5 steps of the glycolysis we can call, friends, preparatory phase. And 6 to 10 steps of the glycolysis, what do we call, friends? We will be calling it as the payoff phase. Friends, please don't get confused with this particular point at all. Now listen my statements correctly. I'll explain in a simple way, but I want you to remember the structures without fail. So I'll go a little faster. Energy investing phase. What we are doing, friends? Glucose. How many carbon glucose? 6 carbon. So the glucose, which is a 6 carbon, converting into what, friends? Glucose 6 phosphate. Friends, glucose 6 phosphate is how many carbon? Undoubtedly it is also a 6 carbon containing compound. But what has happened? Just at the 6th carbon, what carbon? At the 6th carbon, there is a addition of the phosphate group. That is how, friends, the glucose is basically converting into the glucose 6 phosphate. Now understand my words. So someone has to donate the phosphate group. Of course, without someone donate the phosphate group, kinase enzyme can't add the phosphate. So obviously the phosphate group donor will be ultimately ATP molecule. Now once the glucose converted into glucose 6 phosphate converts into what, my friends? It is converting into fructose 6 phosphate. Never ever forget that. Now friends, understand. Fructose 6 phosphate is how many carbon? Fructose 6 phosphate is also a 6 carbon. Now what is happening in the next step, friends? Fructose 6 phosphate converted into fructose 1,6 bisphosphate. And friends, fructose 1,6 bisphosphate is how many carbon? Undoubtedly 6 carbon. There is no change in the carbon number. But what happened? Initially the phosphate group is added at the 6th carbon. Now friends, the phosphate group is also added at the 1st carbon also. Simple point, right? That is what exactly we can call the fructose 1,6 bisphosphate. And friends, this fructose 1,6 bisphosphate, how many carbon? 6 carbon. It is breaking down into 2,3 carbon containing compounds. What are those 2,3 carbon containing compounds? One we can call DHAP, another one we can call G3P. I have told you many times. What is the speciality of this DHAP and G3P? These two are actually acting like a isomers. What, friends? DHAP and G3P both are acting like a isomers. Always remember that point. Now, I really don't know, friends. I want you to remember the structure compulsory. You know, right? This is the glucose. Correct na? What is the ring of the glucose we can call, friends? Pyron ring. Yes or no? How many carbons are there? 1, 2, 3, 4, 5 carbons in the ring and 1 oxygen. What do we call it as? Pyron. So since it's a sugar, we can call it pyranose ring. Easy to remember, friends. Glucose treated by an important enzyme. What is that enzyme? Hexokinase enzyme. It is the one which donates the phosphate group from the ATP hydrolysis. ATP hydrolysis, it will donate the phosphate group. Hexokinase will take that phosphate group and add at the 6th carbon. That's why glucose became glucose 6 phosphate. Now the glucose 6 phosphate, understand my words, it is converting into what, friends? Fructose 6 phosphate, right? So understand my words. There is no change, right? Here 6 carbon, here 6 carbon. What happened? Tell me. Glucose and fructose are what, friends? You all people know that isomerases. Correct? Na? Isomers. So what is the enzyme involved? Undoubtedly, the enzyme involved is the phosphoglucoisomerase, because since it's a both are isomers, it is converting from one isomer to the another isomer. That's all, friends. Now fructose 6 phosphate is a 6 carbon converted into fructose 1,6 bisphosphate. We all people know very well the most important rate limiting enzyme, PFK, which is the part B question which we got in our September 2022 paper. Phosphofructokinase or the PFK. Follow my words correctly. Of course, it is also kinase. It is also has to add the phosphate group. So who is donating the phosphate group once again? Undoubtedly, friends, ATP is the one who is donating this particular phosphate group. Do not get confused with this particular point at all. Right. Now, fructose 6 phosphate converted into fructose 1,6 bisphosphate. Don't confuse. Now, fructose 1,6 bisphosphate is how many carbon? I told you 6 carbon. I want every student has to understand whenever we are breaking something, keep it in your mind, friends. Whenever we are breaking something, we always use the enzyme aldolase. What we use? We use the enzyme aldolase. So fructose 1,6 bisphosphate aldolase is the enzyme breaking down the fructose 1,6 bisphosphate into DHAP and G3P. DHAP and G3P. As a result, what is happening, friends? We break down the 6 carbon into to 3 carbon containing compound. And I have used one important statement. What's the statement I have used? Friends, every single these 3 carbon containing compound are isomeric in nature. What do you mean by that word? Isomeric in nature. Whatever the function performed by the G3P, the same function performed by the DHAP, or vice versa. Whatever the function performed by the DHAP, the same function performed by the G3P also. Don't get confused with that particular point. Now all the students pay attention. The 6 to 10 steps. What do we call 6 to 10 steps? Energy paying off. You only tell me how many ATP we spent in the first 2 steps. We have spent here 1 ATP in the first step. And the third step, we have spent another ATP. Correct na? So total, how many ATP we spent? We spent 2 ATP molecules. Now, whatever the ATP that we spent, we are getting that ATP back. Along with that ATP, we are getting the profit also. Now all the students follow my words correctly. Whenever you are understanding, remember my words, Whenever you are understanding, understand for one compound. Whenever you are understanding, understand for one compound. Whenever you are calculating, calculate for 2 compounds. Then this is the way, to remember it simple. So we got 2 isomers, DHAP, glyceraldehyde 3-phosphate. So I will only consider the glyceraldehyde 3-phosphate, but while calculating, I will add the DHAP and I will calculate. Now glyceraldehyde 3-phosphate, how many carbon it is? 3 carbon. It is reducing. What is the reaction happening here? Reduction. Reduction is happening and it is giving rise to the 1,3-bisphosphoglycerate. Don't get confused. So you all people know, right? It's a reduction reaction. In the reduction, what do we get? We get the reducing equivalent we get. So what I got? I got NADH. Simple point. Now, 1,3-bisphosphoglycerate converting into 3-phosphoglycerate. What do you mean by that? Which means 1,3-bisphosphoglycerate losing its phosphate group. At which carbon? Every student follow me. At first carbon, whatever the phosphate group is present, it loses that phosphate group. Upon losing that phosphate group, 1,3-bisphosphoglycerate converted into 3-phosphoglycerate. So it lost the phosphate. So someone is going to accept that phosphate undoubtedly. Who will accept? ADP will accept, combined with PI, it gives rise to the ATP. Right. Now friends, please don't get confused at the 8th step. 3-phosphoglycerate converted into 2-phosphoglycerate. What do you mean by that? Is there any lose or gaining of the phosphate group? No way. Here there is no loss of phosphate group, no gain of phosphate group. What happened? There is a change in the position. What happened? There is a change in the position. 3-phosphoglycerate converted into the 2-phosphoglycerate, which means, remember everyone, phosphate group, follow my words, phosphate group from the 3rd carbon shifted to the 2nd carbon. That is the simple reaction happened. Phosphate group from the 3rd carbon moved to the 2nd carbon. So 3-phosphoglycerate converted into 2-phosphoglycerate. No loss, no gain of phosphate group. Now the 2-phosphoglycerate finally converted into what? Phosphoenol pyruvate. 2-phosphoglycerate is how many carbon? 3 carbon. Phosphoenol pyruvate is how many carbon? 3 carbon. There is no change in the carbon number. Phosphoenol pyruvate finally converted into pyruvate. By pyruvate is how many carbon? 3 carbon only. Then what happened? Is there any loss of phosphate? Yes. From the phosphoenol pyruvate, there is a loss of phosphate group. So as a result, what is happening, friends? That phosphate also accepted by ADP, combined with PI, and it can get the ATP. So far, I have explained only with respect to one, that is the glyceraldehyde 3-phosphate. But while calculating, we have to calculate compulsory with this. So how many NADH I get? I get 2 NADH. I get 2 ATP molecules. Correct or not, friends? Please don't, I will get the 4 ATP molecules. Yes or no? In the 7th step, I will get 2 ATP for 2 molecules. In the 10th step also, I'll get the 2 ATP for the 2 molecules. Don't confuse. And is there any necessity to remember the structure and enzymes? Compulsory. glyceraldehyde 3-phosphate. I told you now sixth step. What is the sixth step? I told to all of you friends, the sixth step is the reduction. So obviously the enzyme will be what? Dehydrogenase enzyme. Please friends, don't get confused with this particular point at all. So, and glyceraldehyde 3-phosphate giving rise to the 1,3-bisphosphoglycerate by the production of 2-NADH. 1,3-bisphosphoglycerate, what it is doing? It is losing one phosphate. Upon losing one phosphate, it get a 3-phosphoglycerate. This phosphate group lost. Who will take that phosphate group? ADP. Combined with PI, it get ATP. Now remember, I told you now friends, in the 8th step, just the phosphate group is shifting from third carbon to the second carbon. Nothing has happened friends, is it or not? So what will be the enzyme that is required here? The enzyme required is the phosphoglycerate mutase. What is the name of the enzyme friends? The name of the enzyme is phosphoglycerate mutase is the enzyme. Right. So understand my words correctly. Just shifting of the phosphate occur. Then the 2-phosphoglycerate converted into phosphoenol pyruvate. Then the phosphoenol pyruvate upon losing the phosphate group converted into pyruvate. So friends, what is the overall reactions now? All the students follow me the simple calculations. In the step 1 and step 3, we spent 2 ATP. Is it? And in the step 7 and step 10, we gained 4 ATP. Okay. So since we spent 2 ATP, we are subtracting the 2 ATP. Our net ATP is total 2. But in the 6th step, we also got 2 NADH molecule. So 2 NADH giving rise to 6 ATP. So 6 plus 2, 8. So every student remember, when they ask you gross ATP in the glycolysis, which is always 8 ATP, gross. But in the glycolysis, if they asked you what is the net ATP, always remember net will be the 2. Gross means total. Net means how many visible. Correct na? How many are visible to your eye. That is called net. Net is only 2 you can see. Which you can't see directly is this 6, right? So but total we have to consider. Total is 8. Net is 2. Don't get confused with that. Okay. So I hope everyone understands the concept clearly. Please revise this once again. We'll meet in the 3:30 p.m. class and we will finish immediately the process of regulation, most importantly. Okay. Is it clear everyone? Requesting all of you revise this and attend the 3:30 p.m. class. We will finish it off. Hope it's clear. I think we went little faster, but that's okay, but because you are good with the concept already. Clear? Okay friends. Okay. Thank you all. Thank you. I'll see you